Can you see me if I stand here? Yes. What about now? Yeah, okay. I think I can stand at the podium if that's okay.
Well, yeah, no, do your thing.
Okay. Hi everyone, my name is Kieran Samuk. I'm a postdoc with Mohammed Noor at Duke. And today I'm going to tell you guys about a project we've been working on looking at adaptive evolution of recombination rate in Drosophila pseudoobscura. Okay, so just to orient everyone, recombination is the exchange of genetic material that occurs between homologous chromosomes. And so this is just one type of recombination crossing over. So we have our two homologous chromosomes, red and blue. In this case, one sister chromatid of each homologous chromosome crosses over, resulting in the exchange of genetic material. So everyone should be pretty familiar with that. Recombination rate is simply the average number of, for example, crossovers that occur in a genomic interval each generation. So if we again imagine our homologous chromosomes up here, so now we're just showing one line for each, red and blue. In an individual, they undergo during meiosis, there's many possible configurations of gametes that can be generated. And we can basically just, for example, if we want to calculate a recombination rate for a chromosome, we can just count up the number of crossovers that occur, 2 here, 1 here, 0 here, etc. Take an average of those, and that's our recombination rate. So recombination rate is pretty cool for lots of reasons. One is that there's enormous variation in recombination rate across the tree of life. So this is a really cool figure, love this figure, from this really great review by Stapley et al. From 2017. And so this is showing recombination rate on the y-axis and then all kinds of different tasks on the x-axis. So one thing about this is a log scale here. So this is like some pretty crazy variation we're seeing in recombination rate, both within and between taxa here. So that's pretty cool. Another really fascinating thing about recombination rate is that it's a key modulator of all kinds of different evolutionary processes. So there's all kinds of different examples of this. Recombination can impede or facilitate adaptation. We also see associations between suppression of recombination rate during speciation, for example. This is also like with sex chromosomes, as we just heard about. Recombination rate also, we're still kind of spinning up this literature a little bit, but recombination rate also appears to have important consequences for where gene flow can happen and where divergence can happen in the genome. So recombination rate is important and it varies a lot, but we still have a fairly poor understanding of the evolutionary mechanisms and genetic basis of variation in recombination rate. And so today I'm going to talk to you about 3 kind of questions that we asked in our system to try to increase our understanding of recombination rate. So the first was, how does recombination rate vary within and between populations? So we're trying to basically establish like basic evolutionary biology of recombination rate. What genetic changes underlie differences in recombination rate? And are these differences, if any, driven by selection or drift? And so our study species that we do a lot of our work in the Noor lab in is Drosophila pseudoobscura. This is a species of Drosophila native to mostly Western North America. And we focused on populations from 2 sites, Madeira Canyon, Arizona, and American Fork Canyon, Utah. So Madeira Canyon is a sort of desert habitat, whereas American Fork Canyon is a little bit more high altitude. So why these particular populations? First of all, there's not any large differences in chromosomal arrangement, like large segregating inversions. We didn't really have to deal with the differences that may be caused by those, although those are probably important modulators of recombination in natural populations. They also have different environments, so there's scope for divergent selection. There's also some gene flow, which it will be important for looking for selection, which we'll go into later. Okay, so the first step here was making collections at all these sites. And so these collections were actually made by Katie Ritz, who did a master's degree in the Noor lab. And so Katie made collections at both of these sites and created inbred lines. So for this study, we'll be talking about 7 inbred lines from Arizona and 10 from Utah. And so kind of the first step here was we did whole genome sequencing on all of our inbred lines, and we also had a reference line, which is just a reference genome line. So we knew everyone's genotype. And then with the help of Freddie Hsu and Brendan Winkler in the Noor lab, we created these backcross-like mapping populations. So basically the idea here is we take our inbred lines, we cross them to our reference line, that gives us F1s, and then we back cross those F1s to whatever their grandparental or their parental inbred line was. And this generates all our back cross offspring. And so the idea here is, oops, there we go. So the idea here is we have our all these back cross individuals, and we're basically going to genotype them at known markers that are diagnostic of either the reference line or the inbred isolines. We're going to genotype them along the chromosome, and we're going to look for places where we see switches in homozygosity. So you can see here in a homozygous region, there's no crossing over, and when crossing over happens, there's a heterozygous region. So we're basically trying to infer the haplotypes here from these genotypes on the basis of our cross design. And the way that we actually did this genotyping was using this amplicon sequencing approach. So we had 8,000 backcross individuals total. We had about 100 diagnostic SNPs per chromosome. And so we're actually able to sequence all of these individuals in one lane of, or 2 lanes of Illumina, all 8,000 individuals. So that's pretty cool. And it's using this method called GTseq, which is developed by this guy named Campbell in Sean Aaron's lab. Okay, so back to our questions. So first, let's talk about how recombination rate varies within and between populations. So just to spoil it, it does. It varies within and between populations. and here's what that looks like. So on the y-axis, we have recombination rate here. And then our chromosomes on the x-axis. So these are all the chromosomes of Drosophila pseudoobscura. We have our Utah population in red and our Arizona population in blue. And so first thing to notice is there's quite a lot of variation within and between populations. So even like, for example, looking between these red points here, lots of variation there. What's cool is we can look at population level averages. So these are lines within population, but we can switch that down into population level averages. And here's what it looks like. So basically, the Utah populations have approximately 10 to 15% higher recombination on most of their chromosomes. So this is looking at the chromosome scale. We can also look at that locally along the chromosome. So this is now just for the X chromosome, Drosophila pseudoobscura. We have our crossover rate on the Y, chromosome position on the X. And so this is just showing the rates for Utah. So lots of variation locally. And what's cool is we can overlay our Arizona line on top of that, and you can see there's all kinds of interesting places where, for example, Utah is higher, or where it might be the same or lower. So this extended population difference is actually varying across the chromosome, which is pretty neat. Another interesting finding that came out of that is there's a correlation in recombination rate among chromosomes within a cross. So this plot showing basically if you have a high recombination rate on XR, you also have a high recombination rate on chromosome 3. And this is true across all the chromosomes. And we think that this correlation might be implying that there's genetic variation for genome-wide modifiers of recombination rate. And we're doing more to explore that, but this is kind of a pretty compelling correlation. Okay, so just to sum that section up, we found the recombination rate varies extensively within and between Drosophila pseudoobscura populations. Utah has approximately 10% higher crossover rate, and we're seeing this variation at local and global scales. Okay, so I'll just quickly touch on the other aspects of recombination that we looked at. So what genetic changes might be underlying differences in recombination? So we kind of took 2 approaches to this. The first was looking at candidate genes in the meiosis pathway, or pathways associated with meiosis, and also looking at structural variation. I won't talk about the structural variation stuff today. Basically, the idea here is things like large inversions or undetected inversions or potentially deletions. Any kind of region of non-homology that exists between the tester line and our isolines could be contributing to this. We did a bunch of PacBio sequencing to look at that, but basically we found no effect of any structural variation. But I can talk to you guys about that after if you're interested. Okay, so I'll quickly just touch on this candidate gene stuff we did. So basically the idea was we wanted to associate allelic variation in candidate genes in our isolines with recombination rate that we observed. And so we went to the literature. We pulled out 50 strong candidate genes associated with meiosis. We went to databases, pulled out the coding sequence for each one of these. So we're only looking at coding changes in this case. Then we got our genotypes for our whole genome sequencing, and then we basically did a sort of baby GWAS where we analyzed association between these candidate genes and recombination rate. So here are the two genes that had a significant association with recombination rate. So we have our crossover rate on Y, genotype in the coding sequence on the X. So these are both, we're only seeing the homozygotes here because these are in red lines. But basically, these have fairly large effects on genome-wide recombination rate, each one of these alleles. So they happen to be in any sort of opposite direction with respect to the reference genotype, but they both have quite large effects. So we think these are potentially likely candidates for these genome-wide regulators that we were seeing before, which is pretty neat. Okay, so back to our questions. Finally, I'm just going to quickly talk about this kind of cool approach that we took to looking for adaptive differences in recombination rate. So the approach we took was sort of what is classically in quantitative genetics. A lot of you might be familiar with it. It's called the QST-FST approach. And the basic idea behind this analysis is to ask the question, are population differences in a quantitative trait greater than you would expect under neutrality or some neutral model? And so the basic principle is you get some distribution of FSTs from genetic markers, and those are sort of represent your neutral expectation of differentiation. And then you compute a QST value for your quantitative trait. So in this case, this would be for recombination rate. And you're comparing your QST values to this FST distribution. So it's like an FST outlier test, but the quantitative trait. So we did some RAD-seq to generate neutral markers from wild individuals. So these are not from the inbred lines, actually. So you can see that FST is really low on average. The average is like 0.01. So we have this peak, actually peaks in the negatives, and then long tail going out. So just to jog your memory here, this is that difference in recombination rate that we observed at the chromosome level. And so the idea was to compute a QST for each one of these, and then map those back onto the FST distribution. So here's our FST distribution. Here's, for example, the 95th percentile, and we can kind of consider things that are above the 95th percentile, extreme, or unexpected under a neutral distribution. There's more formal ways of doing this. So here is what it looks like. So we mapped on each one of our chromosome estimates of QST onto that FST distribution. And so you can see that these X and chromosome 2 don't really have particularly high chromosome level recombination rates with respect to the neutral distribution, but genome-wide and also on 3 and 4, we observe much more difference in recombination rate than we would expect here. So we're still doing work on this, but we think this is kind of an interesting approach that other people might want to use for studying differences in recombination rate. And in our particular case, we think this also might be showing evidence of local adaptation in recombination rate, with Arizona being smaller than Utah. So just to sum up, we found that recombination rate varies within and between populations. The differences that we found are associated with variants in meiosis genes, and particularly there might be this implication of the local regulators of recombination segregating. And finally, we think that we have kind of interesting preliminary evidence of recombination rate showing evidence of natural selection. Okay, and that's all, and I'll take questions. Thanks very much. Yep.
So, how would you expect your differences in recombination rate to affect your neutral expectation for FST?
So like if differences, like the population level differences in recombination rate, I don't think there's any reason to expect recombination rate per se at genome-wide level would affect FST, but I'm actually not 100% sure about that. It's a good question. Yeah.
Have you looked at the distribution of tenomere keys along the chromosome and see if any correlation with the recombination rate?
Yeah, so we actually did I didn't include this in my talk, we did a whole analysis of looking at TEs and repetitive elements, and we didn't find any evidence that they're correlated with the recombination rate in this particular cross. So these are pretty closely related, so there might not be many differences. That might be one reason, but yeah, it's good. We also have the same thought. Yeah.
If the factors affecting recombination rate are genome-wide, what's going on with that one arm of the X that showed no difference?
Yeah, that's we're not sure. That's a really good observation, but yeah, we're hoping to look more about that. It might be a detection issue. Thanks everyone.